In this video we will cover one of the application of the RIBOmap that is being presented on the paper is to map the mouse brain tissue. So what they did is they analyzed the 5413 gene that they curated from the literatures, and this part of the these genes are belongs to this part of the mouse brain that is being shown here. And this panel B shows the images of these 5413 genes that are being fluoresced through with the help of RIBOmap strategy. And the red triangle that some of you can see, the zoom image of this rectangle is shown here to present the individual cells, how the individual cells looks like. And these images on the z-axis present the different channels, or we can say present the different frame of the cells, so that we can see the difference between the genes that are available in the z-axis. So next, what they did is they analyze this RIBOmap and compare it, analyze the mouse brain of the RIBOmap with the help of RIBOmap, and compare it with the literature data that is presented in the Allen Brain ISH Institute. And they can see that RIBOmap is able to replicate to the data that is available in the literature to a very great extent, and they did it for three different cases that is presented here, and they are almost similar to the data that is available in the literature. Later on, what they did is they did the clustering of the genes. They did two types of cluster. First one is the major cluster, and second one is the subclusters. In subcluster, they classify the genes on 38 subcategories, and major cluster they classify them on around 10 categories. And they are able to see that these categories is highly in correlation with the data presented, or data that are available or published in the past. Thereafter, later on, what they did is they try to map this part of the brain on the with the help of RIBOmap, and they can see that the resemblance between the mapping that is done by RIBOmap with the data that is available in the literatures are highly in correlation with each other. And later on, they presented the data for some specific part or some specific genes in the panel G, and in order to depict their specificity or accuracy of the RIBOmap, we can see that each part is in resemblance with the data that is available in the literature for the like this T, PN, DE, MEN, CHO, VAS, OLG, and AC. And they did this experiment the two times, and all the two times that too in the biological replicate, and they find out that the correlation between both the experiment is very similar, is very high. uh, this predicts that this indicates that the data generated by the RIBOmap have a high accuracy and are reproducible. So this is one of the application that is presented in the paper, and which shows that the superiority of the RIBOmap against the already published translator method, that is star map. And one thing that I forgot to mention is the map that they have shown here in the panel D and panel D. Panel D and panel E is based on the UMAP generation. They cluster data based on UMAP clustering for both panel D and panel E, and then compare the groups through from the data that is available in the literature. So I hope you have understood this one of the application of this RIBOmap and how useful it is. And if you need to understand these things or this experiment in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.